For the next 25 minutes plus questions, I'm not the chair. I'm a very excited principal investigator. Going to present to you the initial report from the Crest clinical trial. And for those of you that run clinical trials, you know the commitment that it takes to run a trial in a double-blind, placebo-controlled fashion, and all the elements within in terms of the quality control, randomization, data safety and monitoring committee. And that's what I'm happy to present today. It's a very, very new piece of analysis because the trial actually only closed in June. So this is the first cut in terms of what we've learned from the data that's available to you. So I'm going to try and take you on the journey of the Crest project over the next 25 minutes. I don't really need to go through the background to this audience because it's this audience that educated me on why we should study this yellow filter in the back of the eye. And we've been looking at this over the years from an AMD perspective, also from a visual performance perspective, and how those are interlinked. And essentially the opportunity is that we try and figure out how to enrich this central yellow filter in the eye in a way that we believe is good for visual function and healthy for people maybe at risk of macular degeneration, etc. And what we've learned over the years is that it's a very variable pigment, and that in this room alone, I can guarantee you would have extremes from maybe close to 0 to a log unit of macular pigment. So it's something that varies dramatically between us. And I suppose our contribution, all of us, to this field was to figure out what it is and how we can try and optimize that using nutrition, using supplements, etc. And the implications of that, and we see from Randy Hammond's lab, from the excellent work that they've done over the years, the implications for visual function and how this nutrient extends beyond that of age-related macular degeneration and into the world of visual function, visual performance. And these are just some pictures to remind us of the opportunity that this pigment presents if we can enrich it. And all of us have contributed greatly to the peer-reviewed science. Many trials, and ourselves we've run many single-blind trials, but also we've done some double-blind trials with a product called Ocuvite lutein. But really for me, the Crest project, which I'm about to present now, is kind of the best of the idea that I could take from all the work that's done and put into something very robust and very secure in terms of how we test this research question. And we've learned so much today about Alzheimer's disease, the brain, the work from Liz Johnson's group in terms, and Neil Kraft in terms of where the carotenoid nutrients are located in the brain. And we've learned a lot today about how that potentially impacts on cognitive function, and it essentially appears to be very interesting associations in play there. And so the publication by others, as I've referred to here, this isn't very, very new to a lot of people in this room. We have been talking about this for some time, and you can see some of the publications. And this morning from Professor Mulcahy's lecture, we've seen that in Waterford we've been looking at this with support from the Howard Foundation to do various trials called the CARDS clinical trials. But the project that I really want to get onto is the European Research Council funded trial. And I purposely put up the finance on it because to run a clinical trial of this nature, this is the type of fund that we're talking, to do it at the level where you engage with pharmacy, where you engage with outside statistical consulting, where you engage with all the various experts that you need to do to test the research question in totality. And that's really what we tried to do with best efforts in the Crest clinical trial. And of course, the best efforts here were to publish the methodology that we wanted to use in this clinical trial, and this has been available in the peer-reviewed literature for some time now in terms of our idea behind Crest, what we were trying to achieve, and the methodology that we used that was informed by the science in full. And this is published by our PhD student, Kwejo Akofu, who's here with us today. Crest had two main research questions. In trial 1, the question was, does supplementation with all 3 macular carotenoids in the 10-10-2 ratio, this is the MacuShield formula essentially, or in the US it's MacuHealth formula, does that impact on visual function in normal subjects without retinal disease when compared to placebo? And trial 2, which I'm not going to present today, but just to let you know that this is what's going to come for the future, was essentially a head-to-head trial with the ARIDS-2 formulation plus the ARIDS-2 formulation including mesozeaxanthin. I don't have time to go into the design of that trial today, and there's no point because I don't have the data, but just to say that there was 2 really clear research questions within this. One was in that AMD group, and the other is in the subjects, healthy subjects, by definition, no visual abnormalities, but they had a unique recruitment strategy, and that was they had low macular pigment levels to be entered into the trial. That's one of the criteria that we went with for that trial. And this just horrible busy-looking slide showcases that, this is a project supported by the essentially European government, the European Research Council. We used all the data that we've been collecting over the years to justify, to win the funds to do this trial, and that was all our work with the most trials with mesozeaxanthin, because we used those exploratory trials to justify to the reviewing committee that they needed to invest in a proper double-blind placebo-controlled trial. And we had many, many contributing services and experts that made this trial possible, and I won't have time to thank them all or acknowledge them all here today, but we went to every effort possible to bring in all elements here. It was a 1-year placebo-controlled double-blind interventional study, and the active group, as I said, was the 10-10-2, and that was versus a placebo. The methodology. So, as I said, one of the things that we wanted to achieve was, over the years, we were looking at the idea of people with low macular pigment. Are they the most likely to respond to a supplement? So we made a decision based on previous studies, and it was actually a recommendation from Dr. Lockman, who's with us, that really when you want to look at visual function, we felt that taking people with suboptimal, I think Liz used the expression of suboptimal levels of carotenoid, we wanted to represent that by looking at people with suboptimal, what we defined as low macular pigment. The idea was we would randomize 120 subjects into either the active or the placebo intervention, and essentially we would do clinical assessments throughout the trial at baseline, 3 months, 6 months, and at 12 months. This is some of the eligibility criteria here. Best corrected visual acuity of 6 or better, so they had good vision to begin with here. No previous consumption of supplements containing the macular carotenoids. In the Crest trial global, we went to massive efforts to make sure that we limited contamination in terms of confounding variables at baseline. And for example, we wouldn't allow subjects into the trial if they were already taking a supplement. And if you extend that to our AMD project, we went to massive lengths to make sure that they hadn't previously been taking supplements, or that the disease that they had in their eye was in fact macular degeneration and not many other diseases. But as I said, we were saying normal here in that they had no vision-related abnormalities, and this was an important recruitment strategy. Now, what we did was we had to screen over 400 people from the population to get our people with our, as defined, relatively low macular pigment. And the data I'm going to show today is from the Spectralis, because it's the data that we have available for analysis at this point. And this is just an example of some of the low macular pigment subjects. And you can see that these subjects, we didn't see any trend towards whether they had atypical profiles. You can have low pigment, as measured on this device, and have various different spatial profiles, as you can see here. How did we do it? It was a massive marketing campaign to advertise this trial to the local population, and we were very successful dealing with even TV nationwide productions where we spoke about the trial, where we spoke about the importance of the trial, and we also used local newspapers and radios to advertise the trial, and that's how people from the Waterford and surrounding population came for that screening eligibility visit. The primary outcome measure was contrast sensitivity at 6 cycles per degree, and this was informed from a couple of previous papers that we had published, some from our most trials, but also we saw very clearly in our cataract work that a measure of visual function that we believe corresponds well to subjective visual function is contrast sensitivity, and we see that at 6 cycles per degree, this was a measure that had a lot of hits in those previous trials. In other words, in the cataract trial that I'm speaking about, the subjects that, when they had the cataract surgery, that they said, okay, I feel better, the measure that corresponded best with that was this contrast sensitivity measure. So there seemed to be a trend coming out that this was a measure of visual function that would be sensible to look at. We had many secondary outcome measures, macular pigment, glare disability, cognitive function was what I described in the grant proposal as a new horizon section. It's something that we should be looking at. And this was obviously down to the work from Liz Johnson and others that kind of has guided us in this direction, that we should be looking maybe at these other measures. So luckily we got that into the study protocol many years ago now. And there's some of the other measures, serum carotenoids, foveal architecture. As I said, we had baseline 3 months, 6 months, and 12-month study visits, and it was very interesting to listen to Lisa speak today about the many things we can do, and I suppose the overall equation and package of assessment one should do if they want to, for example, look at cognitive function. I think that's brilliant, and I think we need to learn from what Lisa has taught us today. But I also think it's, from a clinical trial point of view, we need to look at this in the context of what's feasible in say, a 2-hour period, and we have to take fatigue and everything into consideration when we run a clinical trial of this nature. Particularly, for example, if cognitive function is just a secondary outcome measure, we can't spend an hour of our assessment time doing the very nice test that Lisa referred to. But we did take advice from experts in this field, such as Robert Cohen, who you heard speak today, on what battery of cognitive assessments at that end stage, as I've learned today from Lisa, that's really what we were assessing. What battery of tests should we look at if we want to see if there's a relationship between an active intervention? We do all our work in-house. We do everything from our analytical work up to our visual assessment work and our cognitive function work. It's all done in-house in this controlled environment, and this is just a photograph of our laboratory, and Rachel Moran, who did a lot of the analytical work, and Dr. Dave Kelly, who did a lot of the analytical work on this. This is the laboratory they will work in and work on this data. We have all our own -70 freezers where the bloods are put straight away once they're taken from the subject. And we're familiar with the extraction protocol. We've presented this before and we've published this in detail elsewhere. But we have a very nice method now for allowing us to look at lutein, zeaxanthin, and mesozeaxanthin response. We use, for this analysis, we use the 2-step method as presented by David at the last conference. And just to give you just to show you, like, here we can see the red line, and this is courtesy of Rachel, again, this chromatograph. You can see that an unsupplemented subject sits here. But when you give them, this is obviously the MacuShield supplement, you can see that you get this massive response very quickly in blood, 500%, 600% response in blood. So what that tells us in terms of carotenoids is that if you give people a supplement, on average, and typically they respond very, very well to these particular nutrients. And here you can see an example of our mesozeaxanthin peak, which we use and work back using our method of proportions. As I said, today I'm limited to presenting the autofluorescence data. We were the first people in Europe, I believe, to have access to the macular pigment component of the Spectralis device, and we're familiar how that works. It's a blue and a green, and we basically use the nice software that's provided by the Spectralis to give us this lovely image of macular pigment. That's half our macular pigment mountain, which we're presenting there. So it's nice to have an image, and some of the advice that Paul Bernstein gave me when he realised we were starting this big venture was to using imaging technique in addition to our Flickr device, and we took that on board and went at lengths to secure this technology. We measured contrast sensitivity in many different ways. Thomson letter chart was the main measurement of contrast sensitivity that we used in our vision laboratory, but we also dabbled in the functional acuity analyzer, which has options for kind of daytime, nighttime conditions as well. We use the battery, and Robert Cohen explained it to us today in terms of how we assess cognitive function. And importantly, I'll say that specifically for this trial, Robert Cohen advised and worked with Cantab to make tests of cognitive function that were sensitive enough, in theory at least, to what we were trying to do in this healthy, younger population. So we weren't, like you made the point, Lisa, about MEMSI, all these guys are going to score very well on such crude assessment techniques as that. So it was customized to meet the criteria for this study. And essentially, we're measuring memory, we're measuring reaction, we're measuring all measures of cognitive ability in this trial. Statistical methodology, for me, was very important to this trial, and how we approached it. And Dr. Jim Stack, who was employed into this project as a private consultant to do advise on the statistical power and also to publish the statistical analysis methodology that we're allowed to use for this trial, working closely with the DSMC. as I said, we luckily had access to previous trials to understand means and standard deviations of, say, contrast sensitivity to allow us work up or have suggestion around power for the study. And this was all advised by Dr. Jim Stack. Another thing I'll point out for those of you that are interested in statistics, we use block randomization, which for a relatively small sample trial, which this is, let me be clear, block randomization I believe is important, because sometimes with just normal randomization you can have a big split between your active and your placebo, but block randomization essentially assures that you're going to get relatively equal numbers in each group. And the method that we published before the trial data was available was that for baseline assessment, for interval analysis, we'll use independent sample t-testing, and for we would use categorical variables, we would do the chi-squared contingency assessment. And the change over time is basically, for the non-statisticians, it's quite simply, it's a repeated measures test, so we can look at different time points. And the research question within that type of analysis is, do the two groups that are in the trial differ with respect to the measure of interest? So it's not, sometimes you can do basic t-tests and you can take a group and look at them and see are they different from the beginning till the end. This would be a much stronger statistical approach in terms of how we address that question. In other words, you may with t-tests see significant differences from baseline to end which wouldn't survive in this type of analysis. And here are the results. The recruitment history, we began recruitment on the 2nd of October 2012, and the last subject was recruited on the 3rd of June 2014. So it took much longer than we wanted or anticipated to get the subjects to meet the inclusion criteria. And one reason for that, and let me make this point, is when you're studying AMD patients, you're studying the well-worried. You know, they have a reason to want to be in the trial, they have a reason to want to come back. It's very difficult to explain to a 35-year-old male that, you know, he should do something with nutrition or a supplement that may be good for his or her eye. And that is an issue for a clinical trial of this nature. You know, the attrition rate that we've seen in previous trials. So we had to go to extreme lengths to support the subjects that were coming into this trial, to keep them and to ensure that they comply to the study interventions. And as I made the point already, we screened over 400 subjects to get the sample that we recruited. Subjects deemed eligible at baseline was 95, so therefore we didn't meet the required or the needed sample size. Needed is the wrong word. We didn't meet the sample size that we had hoped to get. But you'll see later that it doesn't matter in terms of our power. From 0 to, we looked at compliance, because I think this is very important for the reasons I've mentioned about, you know, the well-worried, and in these subjects, by definition, we're not well-worried. So the risk to this study was that they may stop taking the supplement. They may just lose interest in the trial. So we did a lot of work counting tablets and compliance and so on. The conclusion from our assessment is that while there was some differences from baseline to 6 months, and from 6 months to 12 months in terms of number of tablets missed on average, it wasn't significantly different between those time points. And that was some way encouraging to see that the subjects probably continued to comply in a way. The other thing I will say is I truly believe that you have different levels of compliance within a population like this. I think you'll have some people who get up in the morning and take their supplement every day. You'll have other people who don't comply as well as that. So I think when we interpret the data from these clinical trials, you have to keep that in mind. And that makes it difficult when we want to speak about non-responders or poor responders. I think that is something we need to keep in mind. We also use our serum carotenoid assessment at the end of a study as a way to help us understand compliance to the interventions. At baseline, and this is the result of the very elegant randomization process we use, we can see here that the measures that we were interested in collecting, that at baseline we had a beautifully squeaky clean comparison in terms of the two groups. They were identical in every which way. And I can tell you that based on other trials I've been involved in single blind, this doesn't happen. And that can cause problems when we're trying to do the analysis and control for things like age, etc. So this is the result of going to the efforts that we did to have this elegant study protocol. And here you can see all the other measures of macular pigment, contrast sensitivity, and so on. And here, these are the cognitive function measures which we heard Dr. Cohen speaking about today. And the subjects in this trial were comparable with the exception of synaptic fluency, and our statistician therefore would have to, if we're looking at this particular variable, we'd be aware that they differed at baseline. Just an observation, and I really don't want to distract my talk by going into this. because this is not the main point of the talk, but when we were looking at the serums, we did notice that there was 3 subjects in this trial that at baseline had a peak that for us looked very similar to the mesozeaxanthin HPLC peak that we see here. And Alfonso and David Kelly went back and checked this for me and did confirm that it appears to be a peak in the serum before they went on supplementation that looks like mesozeaxanthin. So what we did was we contacted the 3 subjects in question and we got the following information. One is that the first question we asked them was to be very sure to confirm that as far as they were aware, they hadn't taken a carotenoid supplement. And 2 of them said absolutely with certainty that that was the case. The other person said maybe 12 months ago or 2 years ago they would have taken one of their MAM's MacuShield supplements. So for that reason, the lucky point is that this subject actually was a dropout, so therefore is not included in the analysis anyway. But I just think it's an interesting observation. The other, I queried directly to them on the diet. Was there anything specific from their diet that may account for this, albeit small concentration of mesozeaxanthin? And 2 of them independently said that they eat a lot of tuna, referring to tin tuna as a potential source, I believe, of very, very small trace amounts of mesozeaxanthin. And speaking with Alfonso, we believe that it could be even the processing and the packaging of the tin tuna, the process it goes through to get it to its final selling point. But again, not to take from the trial. The main results I'm going to show here now are the response that we got in serum. And we've had a lot of conversation over the last few days about, diet versus supplements versus eggs and which is better. What I'll say is that in the context of this trial, you can see very clearly here that the subjects on the active intervention give a very, very significant response to each of the carotenoids of interest within that formulation. And this is the MacuHealth or the MacuShield formulation. And again, down to the nice design of the trial, you can see a very straight placebo, which is nice, and you see your active response. I will point out that there appears to be a drop from 6 to 12 months in terms of serum, but this is something I've seen before in other studies. And for me, some of it could be compliance dropping, as we've discussed, but another reason I believe it's probably more likely that the tissues around the body are probably starting to soak this stuff up, or it may be linked to binding proteins becoming available. I don't know. I'd like to hear comment on that from the audience. But this is a trend I've been seeing in many of our interventional trials over the years. In terms of macular pigment, we get a really nice response and a continued response, similar to what we've seen in our single-blind trials. But the quick answer here is that the supplement worked very, very well to change and augment and enrich the pigment as we had hoped it would do. I will point out that we did a very similar trial using an Ocuvite lutein supplement, the Compass trial, many years ago. And at 12 months, we got nowhere near this type of response for whatever reason, maybe the supplement, the way it was packaged compared to what we've been able to achieve with the 10-10-2 formula. Change over time, just to give you some examples, you can see this is an example of a subject with 0.37, and at the end of the trial, that's 0.62 optical density units. So this is the type of response we were seeing individual subjects in the trial. So this, basically, we have greatly enriched the amount of yellow macular pigment in the eye with this supplement. In terms of the contrast sensitivity, now I only have some data to present today because of analysis time, but we can see here that the primary outcome measure actually, with repeated measures analysis, shows a statistically significant improvement in contrast sensitivity. There was 2 hits for contrast sensitivity. One was at 1.2 cycles per degree, and the other was at 6 cycles per degree, which was in fact our primary measure. And just to show you how we measured, this is Rebecca, who was, by the way, excellent in helping me get this report ready for you today. She worked very hard with me to do this. And you can see here that this is the type of, this is for 1.2 cycles per degree using the Thompson chart. And essentially, the contrast fades to a point where they can't see it anymore, and we're able to get a score of their contrast. And then for the 6 cycles per degree, obviously the letter size is smaller, and again, the contrast fades as you go through the test. In terms of change in cognitive function over time, this was the new horizon. This was something we wanted to try and do. And, we have had some what I believe very important hits here. And we see a pattern emerging from previously published trials to our own cross-sectional work to what this double-blind trial has shown here. Essentially, we've shown with this interventional trial that the 10-10-2 formula gives you improvements in various measures of cognitive function. And the implications of that and what that actually means from a clinical perspective needs to be really worked out. But it's really very encouraging for the data, I have to say, at this point. And remember, this was done with repeated measures analysis, so change over time. The research question was, did the placebo group behave differently to the active group with respect to these measures? And so it's a tough statistical test. And you can see here, in some cases, that quite meaningful improvements have been identified in our active test there. Some other data that I think this audience will be interested in is that if you compare the difference in serum lutein from baseline to the macular pigment difference, we see some nice trends coming out. And this is something that I haven't shown in previous trials in terms of does the amount of macular pigment response you get correlate with the amount of serum response that you get. I hadn't been able to show that in previous trials, even though we know that those two parameters are related. And I think that's probably down to the design of this trial as well, that we were able to pick that up. Another important correlation to show you is that the difference in serum lutein correlates very well, very, very well, in fact, with the difference in mesozeaxanthin or lutein versus serum mesozeaxanthin there. And the point of these correlations, I think, are very important. And that is, there was maybe a concern that having high amounts of meso in the supplement would have suppressed the response of lutein. But overall, we see where people responded very well to lutein, they responded very well to mesozeaxanthin, and they responded very well to zeaxanthin. And I think that's, so is there room for these three carotenoids to coexist in serum in a way that we can enrich the various tissues of interest in the body? This data certainly suggests that is the case. The other thing that we see, I think it goes back to our point on compliance, that I believe subjects complied at different levels to this intervention. So the overall conclusion is that we have achieved with this double-blind placebo-controlled trial a statistically significant improvement in the active group with our previously published primary outcome measure, contrast sensitivity at 6 cycles per degree. We've also identified statistically significant improvements in the active group for the following measures of interest, and that's macular pigment as we had hoped and expected, serum carotenoid concentration, contrast sensitivity, and cognitive function variables. Our job now is to obviously try and publish this important trial, but to really now try and look at the data and try and interpret what this actually means for use, for humans, for health, for clinicians, because I know clinicians like to be able to say there's a trial done that shows X. So how can we translate this very important message to the field? And I believe that, obviously as a community we will continue to understand and work in the area of visual function. But for me, I believe that this kind of highlights my efforts and my team's efforts over the years in terms of trying to understand the impact of these nutrients for visual function. So to thank the Crest team, there's many of them and you see them all there. Everyone contributed greatly. I think sometimes we forget the work that goes into a trial of this nature. People really commit their lives and their livelihoods to delivering on such data. So to all of these people, I'm very, very grateful indeed for their commitment to this work. And of course, the broader Macular Pigment Research Group team, it's really been a community effort for us to do this trial, and we're very proud of it. Our collaborators, again, I don't apologize for the fact that I've used all the good science of the people that I'm looking at in this audience to help us understand and design this clinical trial and to be able to present it in this way. It's a reflection on all the achievements of the very good scientists in this community. So for all of those, thank you very much. And I'm happy now to take any questions that you may have. Thank you.
Very much. We take some questions now.
John, you didn't actually mention that what happens at baseline. Would you like to expand on that in relation to macular pigment and cognition?
Yes, so we discussed this yesterday. This is the David Kelly paper.
Oh, yes, okay.
Yeah, so at cross-sectional level, we've seen that a lot of these measures of cognitive function relate very well, and Lisa commented on this as well. In other words, people with high macular pigment, before any intervention, perform betterly in various measures of cognitive performance, and memory were some of the parameters that came out in that. So at baseline, at cross-section, that's what we've seen. But the real win here is to be able to see what happens if you change something significantly, and I think that's what Crest has achieved so far.
Okay. Question in the back?
I noticed that when you were talking about your stats, you brought up a chart after that, and the placebo group had a lot of variability. And so I'm new to this group, and I don't know if that's normal. I think it was twice what it was in the non-placebo group. Is that correct?
In what parameter?
It was a couple slides after you talked about your statistics.
Yeah, the placebo group was pretty flat in all the measures that we reported, so maybe you misinterpreted the...
Yeah, maybe so.
Yeah.
Sorry.
No, the placebo group, it was very nicely flat, in fact. There was very little... When you do HPLC, it's a very exact science, as you know, and it was pretty much a straight line for serum response. And are you on about the measures of visual function?
This was the standard deviation and the mean. You had a chart with the standard deviation and the mean. The standard deviation was roughly equivalent to the mean.
Yeah, so within both groups, you have variance. So I'd say that's what you saw trending throughout. But that variance didn't change significantly on average.
Okay, I think I'm getting information from the
Okay.
group here that it's compliance was maybe what we were talking about.
Oh,
Yeah.
the compliance data?
Yes.
Okay, yeah. The placebo group did perform poor with compliance in this trial. That is correct. But it wasn't statistically significantly different. But they did perform poor, yes.
I see. Thank you.
Sorry about that.
Just going to make a comment on that, actually. So when we've done intervention studies with micronutrients before, we have seen a lot of variation in the baseline in the placebo group, but actually the active tends to bring that down, which might suggest that some of that variation in the baseline group is due to natural dietary variation.
Yeah.
And so that you can close that up as somebody is supplemented, because the amount that they're getting becomes much more homogeneous.
Yeah, okay.
Okay. John?
Yeah, John, a alternate reason that you might see a drop in the serum levels from 6 to 12 months is your dose level was fairly high.
Yes.
And work from Johannes von Lichtig's group would certainly suggest that the eccentric cleavage enzyme, BCO2, is activated with high lutein, high zeaxanthin. So you may just have turned on a system to degrade.
To deregulate, indeed.
Right.
Okay, cool.
So that's
And there are
highly
also other
likely.
regulatory
Yeah.
ISX is also another one. There's a lot, and we saw that same in our supplementing MACTEL patients. It drops.
It drops, yeah.
In the second year. Yeah.
Okay. And that's interesting because we have a poster today, and in an Alzheimer's group, Kate will be presenting, and we see a very dramatic drop in that particular group. So I'd be interested to have conversation at the poster session on that for sure. Yeah.
John, why did you limit the subjects to -5 refractive error?
Why did we limit, James, can you maybe -5 refractive, why did we limit? Oh yeah, because from the visual function point of view, I think we wanted to kind of restrict it to within that range. So it was just a decision that was made from.
Yeah, it's quite arbitrary.
Yeah.
Where that level is set is not fixed. There's obviously kind of structural differences in terms of kind of very high myopes that might some of the
Yeah.
comparisons we want.
And there's also been publications with high myopia and its impact on macular pigment levels as well. So it was just a recruitment decision.
Okay. We'll take just one more question, probably.
Hi. Any patient-reported outcomes from Crest?
Adverse events?
Any PROMs? Patient-reported outcome measures?
How they feel, is it?
Yes.
Yeah. We have subjective data which we haven't really got a chance to go through in detail yet. But no, nothing that I can report here today at this point. Yeah. Or what I can say we haven't opened the code on the AMD trial, which will be exactly 12 months from now. we have a lot of patients in that trial reporting, lots of positive things. And we've seen that in our previously published trials as well. But we're not sure, obviously, with the code broke, whether they're on which group they're in that AMD trial.
Okay. Well, I'd like to congratulate John and all his coworkers on doing an excellent, well-designed study. That's, this is the way things need to be done.